So I'm particularly interested in the role of soils in the context of climate change. We try to understand whether soils act as a sink for carbon or as a source for carbon. And that it has implications not just for the climate system as a whole, but also for how fertile soils are. So carbon entering the soils and stored in soils is really, really critical for soil fertility as a whole and our ability to grow food.
I study the soil carbon cycle. So soils store more carbon than all of the plants on Earth and the atmosphere combined. And microbes that live in the soil turn soil carbon into carbon dioxide as they're performing microbial respiration. So they're getting energy from that carbon. Soil microbial respiration actually accounts for over 25% of CO2 emissions globally. So any amount of wiggle room on that value can have big impacts on the climate.
The challenge that we have in soils, with them being so complex, having really this intricate mixture of all these different components thrown together and mixed over and over by earthworms and other creatures. This particular microscope is really good at resolving the types of minerals and the types of microbes and the types of organic matter that are assembled in aggregates at really, really small scales. To my knowledge, this microscope is the first one that's really only dedicated to environmental samples. So it's really a great opportunity that we have here at UNIL to explore soils in this novel way and really resolve these intricate interactions between all the different components that we are so interested in soils. The new project of ours is focused on rhizosphere processes, and the rhizosphere is this really narrow zone around plant roots in the soil, where what is happening in the rhizosphere really dictates this balance between carbon coming into the soil and carbon leaving the soil versus carbon being stabilized and sequestered for a long time.
I'm interested in the role of plant roots and how I think that they're basically causing anoxic microsites to form. So for instance, the soil here, we would expect there to be lots of oxygen because it's not flooded, but in reality, there are probably these little pockets that are without oxygen. And those matter for soil carbon because microbes in anoxic microsites perform anaerobic respiration, which is slower than aerobic. So they're not producing CO2 as quickly as microbes living in oxic portions of the soil. So my first project here is basically looking at how particle size and moisture can affect anoxic microsite formation around a model plant root. So we're using a microdialysis probe as a root, and we're mapping the oxygen sort of around our fake root is what I call it. And then hopefully we'll be growing real plants soon.